Hello, everyone. I will now talk a bit about the computational proteomics for post-translational modifications for 40 minutes roughly, and then Carlo will take over and show you how to work with these data in Perseus. So that's the outline for this talk. First, I will explain you what post-translational modifications are. It's a pleasure. I'm a physicist by training, but to explain that. So let's see how that works. But then we come to the spectra of modified peptides. We talk about fragmentation methods, actually how these spectra look like, and what settings you have to set in MaxQuant for these PTMs. Afterwards, we talk about the whole workflow in MaxQuant, like the identification of modified peptides. There are some filtering steps, and then we come to calculation of localization and occupancy or stoichiometry. Okay. So protein phosphorylation is a biological regulatory mechanism. So proteins, they get phosphorylated by a kinase, meaning there is a phosphate group covalently bound to the peptides. And this is then, of course, a very stable bond. And this then actually alters the conformation of the protein, can actually cause activation, deactivation, or otherwise modify its function. It's also reversible, the so-called dephosphorylation, and this is a phosphatase doing this. Here we see protease regulated by phosphorylation. So it's really a lot of processes which are influenced. And there is this histogram, so showing where it's very important. So there's transcription, cell growth, cell cycles, and so on and so on. So it's basically important everywhere in biology. And there is many studies, of course, about that. There is like drug research, identifications of drugs. And we also had this very nice plenary talk on Monday from Judith about Alzheimer's and very impressive PTM data shown there. There is this website, Phosphosite Plus, which offers actually a lot of information, a lot of studies, but also tools about protein post-translational modifications. And this is not only phosphorylation, it's also ubiquitination, somylation, and so on and so forth. Okay, that's all we have to know about PTMs to have a look at the spectra. So in this example, we have an unmodified peptide A, and we compare it to a modified peptide B. And here this star indicates the modification. And as usual, in a MS run, first there is an MS1 scan measured from that. And if we now compare the peaks, the masses of these two peptides, there is a difference. So they are easily distinguishable on the MS1 level because they have a delta mass difference caused by the modification. So in DDA, like these peaks are then selected for fragmentation. They are precursors, and then we get MS2 spectra. And if we now compare these MS2 spectra, it's basically the same. So we see mass shifts of delta mass for many fragment ions. So all the ones here on the right, they are shifted by the same delta. And this is, of course, caused by the fact that the modification is present in these fragments. And the other fragments which don't contain the residue, which was modified, they also have the same mass as before. So from such a spectrum, it's actually easy to find the location of the modification because it's just actually from where on the mass is shifted by delta. Okay, so, but what about the fragmentations? Here we have another peptide, and fragmentation usually is along the peptide backbone. So the ions which contain the N terminus are like A, called A, B, or C. And if the charge is retained on the C terminal, the ion type is X, Y, Z. That we all know. And in the case of modifications, we get actually more information, not only what I showed you, but there's also neutral losses coming from the modifications. And there is also the so-called diagnostic peaks, which is displayed here. So this phosphotyrosine ammonium ion gives us a diagnostic peak. So like really good indicator if the modification is actually present or not. We know there is different fragmentation methods. There is the good old CID, collision-induced dissociation. There is the big sister, which is HCD, and but there's also ETD and there's also others. But let's first have a look at CID and HCD spectra. So they are both producing B and Y ions, but HCD has like this higher energy, right? So it actually has less. So and here we see the sequence, right? And like the present fragments. So it has less of this relatively unstable B ions, but it maintains the Y ions. And there is this one exception which is the A2 that is coming from the actually B2 ion, which is more stable in that case. So this is an exception, but overall the spectra are cleaner with HCD, and also the diagnostic peak can be resolved better because in CID there is like a weird effect which is called the 1/3 rule, which somehow does not allow to actually see the diagnostic peaks. The ETD world is a bit different. So here there is actually electrons transferred from radical anions to the molecules, and that leads then to the fragmentation. But the fragmentation is different. So the ions produced are then C and Z ions, and in general also the charge states are higher of the precursors, but also then of the fragment ions. But overall, it maintains, like it keeps the PTMs and the side chains, and is usable for PTM analysis. I mean, there is a lot of progress. So it's actually this. It said it was slower. I don't know if it's still slower with the like latest generation instruments, but there is development going on, and there's also other fragmentation methods now on the market. But so HCD and ETD can be used. And now let's have a look actually how to set it up. in MaxQuant. So here we see the MaxQuant window, and we are in the. Can you read? Ah, yes, you can. You can read it. I can't read it from here. Okay, you can use that one. No, too short. Sorry. Okay, so this is the group-specific parameters, and there is this section modifications, and you can select your variable modifications from a long list, and also the fixed modifications, and then bring them here to the right to actually select them to activate it for your MaxQuant run. For the real experts. You can also in the configuration tab, there is this list, and you can actually check the settings. You can define new modifications if you want to. There is a YouTube video about that explaining it. So Carlo won't show that. He will only talk about Perseus. But basically, you define it. You can set the amino acids that it's bound to, and then you have settings for the neutral losses that you expect, and also for the diagnostic peak that is to be expected for that PTM. Okay. So let's have a look at the computational pipeline in MaxQuant. So this is we have the fragment spectrum that's like we understood it now, and then it goes into that funnel. So we know that like first is data acquisition, which gives us spectra. We know about this fragmentation methods. You can also think about acquisition strategies, by the way. So some like you can use MaxQuant Live global targeting for that. This is not a problem, or even MaxQuant Live DDA. But anyway, so we have our raw file, and now let's have a look at the pipeline. So first, there is this identification step, similar to what is done in MaxQuant. And then the next step is calculating the phosphosite localization, and afterwards the occupancies. So for the identifications, we can, like, as you learned, try to identify there is a theoretical spectrum needed. And this table shows actually how the theoretical fragment masses are calculated. So here you see the conditions, and here you see the ion series, and this is now getting interesting there. So if you have modifications, right, then you calculate the specific neutral losses. So they should be considered in your theoretical spectrum, and this is the same for the diagnostic peaks. So that just brings additional peaks in your theoretical spectrum, which you can then use. There is this very nice formula that you've seen earlier to calculate a Andromeda score for modified peptides. So when that is calculated, then there is some filtering going on. So there is a minimum score for modified peptides, and there is the minimum delta score for modified peptides, which means that's the difference between the highest score and the highest score for different sequences. And these values are by default set to 40 and 6. And this was actually derived, like all the MaxQuant parameters, again, right? They are well chosen. There was a paper where this was evaluated, which ones are the best. So you should keep them and not change them as long as you really know what you're doing and what you want to achieve. with like changing the parameters, but like stick to the defaults. That makes life much easier. Okay, so let's talk about the localization probability for a site. So this is an example of there's three sites on this peptide. Oh, sorry. 1, 2, 3. Yes. And to calculate the probability is for a site here, you actually the Andromeda scores that we calculated that are used here. So first, for this one, we calculate a P1 by using the Andromeda score as the exponent. And this is done for all of them. So the localization can then be calculated by the sum of these P's for the modified peptides. And this is normalized by with the sum of all of them. And this gives the localization probability. And again, there's parameters. So if this is at least 0.75, then this is actually considered as well localized site. Again, this is like empirical parameters. Okay. So the occupancy is actually the fraction of proteins for which a site is modified. So in this case, we have a set of proteins under a condition A. So this is these 3 blue ones here. And then there is a stimulus in condition B. There is they turn orange. Obviously, and probably there's also a different PTM level there. So let's like have a closer look on this table. So we have these conditions A and B, and we can just like bring these numbers down. So the phosphorylated peptides in case A is 1, then we have unmodified peptides, currently 2. No, yes, it's 2. And in total, we have 3 proteins. And in the case of B, there is more phosphorylated peptides. So, like comparing these absolute numbers is not a good idea. It's actually much better to compare the ratios between it, and that's how it's done in MaxQuant. So, from like these rows, there is the ratios calculated. They're called an X, Y, and Z is the ratio of the proteins. And from using these values, the proportion can, just like so-called proportion A, can be calculated using this formula, and then we get the stoichiometry or occupancy by this formula, which results in a occupancy of 2/3 for our condition B compared to the condition A, or without stimulus. Okay. So in this paper from Kirti, which was a plenary speaker from 14, sorry, that was like everything was presented and actually benchmarked using HeLa S3 cells. And here we have 3 different conditions. So there is the untreated cells, then there is mitotically arrested and released cells, and then there is also stimulated factors by the EGF, the epidermal growth factor. So, and it was investigated if this like label-free algorithm can actually accurately capture signaling dynamics that we expect, right? So, for example, right at the beginning, I showed you that cell cycle, right? There's a lot of phosphorylation going on. And this was then actually confirmed by using the occupancies that we, like, I showed you how to calculate them. So this is the control. It's a histogram over the occupancies of the phosphosides. And in the case of mitosis, so this stimulus, like you can really see like how it's going up and how everything's drastically shifting. And it was found that the key phosphosides, which are known to be activated also in the EGF, they could actually be quantified accurately using this method. And yes, this is good, of course, but it was also like kind of challenging because you need a lot of peptides, right? You need a like big coverage. It's like not with proteins where you like have one or two peptides and then protein is somehow quantified, but this is on the really on the peptide level. So it needs a lot of measurement data, and this was a very deep proteome. So to use it in MaxQuant, you need of course you need your PTM data, but you also need a complementary full proteome. And here in this case, you see that this is like 3 replicates, unmodified, which have the parameter group 0. And then there is the phosphor replicates here, which have which are in parameter group 1, because in that case you have PTMs, and as I showed you before, to set that correctly, that's in the group parameters. So they're different. The experiment name is the same, just to allow MaxQuant to map and do these calculations automatically. And the fractions are set differently. So that is just to make sure that there is no matching between runs happening between these 2 samples, which are, of course, a little bit different. Okay. Yes. And this is also very important. There is this PTM column. So set that to true in case of PTMs. So that's another example here. Just again, we have a deep proteome, but in this case, there is also fractions. So here you can see it's fraction 1 to 5 for the reference proteome, and that repeats 3 times for the replicates. And the PTM data is then here, also like 3 replicates again. Okay, and then you click start, and then you wait a little bit, not too long, and then you get your output tables, which are then handled in Perseus, and Carlo will show you how to proceed, and that's it from the MaxQuant side.